All right, it's about time. So let's start our conference.
Mhm.
Good morning, everyone, to those in the U.S., and good afternoon to those in the U.K. Originally, we were scheduled to have two presentations today, but unfortunately, Brian Bott, who was scheduled to present about Sage Synapse today, was involved in an accident. Our hearts go out to Brian, and we hope for him a speedy recovery. So today we'll have just one presentation, so it'll be a shorter meeting. The presentation will be about BioGPS presented by Chunlei Wu. The presentation will be around 30 minutes or so, and then we can use the remaining time to discuss any updates from the PI meeting, such as like current progress or collaborations. Again, these conferences will be recorded, and after the meeting, they'll be accessible online after we process them. If you have any questions during the presentation, please use the message box provided in the application. And also, please mute your mic during the presentation to prevent any unnecessary sounds. Are there any questions? All right, let's start. So our first presentation will be presented by Chunlei Wu. He received his PhD in biomathematics and biostatistics from University of Texas. He has worked at TSRI for 4 years and is currently an associate professor of molecular medicine. Chunlei also presented about mygene.info a couple of weeks ago, and today he'll be presenting about BioGPS, which is a community-driven, customizable gene annotation portal. Without further ado, allow me to introduce Chunlei Wu.
All right. Thank you, Vincent. It's great to meet many of you guys in the last meeting. And I already presented last time, so you guys probably know. Part of our job, part of our work is about like mygene.info, myvariant.info. This presentation gonna present you another project working in our group called BioGPS. BioGPS is what we emphasize. It is a gene annotation portal, but what we want to emphasize is this is a community-driven and also customizable gene annotation portal, which is how we differentiate BioGPS from the other existing gene annotation portal. Okay, so for BioGPS, the targeted audience we think is all the biologists or bioinformaticians who have done the like a high throughput molecular profiling, and at the end you will get a list of the gene, and this list of the gene can be very long, can be 100, even sometimes it can be like 500, even like 1,000. So then the next task is go through all these genes, trying to find what are the function related to those genes, and particularly for a lot of genes actually is not familiar for the actual researchers. So there's a lot of work you need to do to follow up the genomic studies. So now we have a lot of existing gene portal, gene-specific annotation database to check the gene-specific information. For example, the NCBI entrance gene, and we also have the like MGI for the mouse gene. Also the gene wiki is a very good resources for the gene information in general. And there's many information we can find from the many different, probably hundreds of different resources. That actually become a very long process, and probably a very tedious process for the end users. They need to learn different interface, and they try to figure out how to use all the different websites. all this information, when you go to this website, you can find that most of the information actually quite similar. Maybe all of this website will have the gene name, gene symbol, like a ref-seq, maybe gene function, but almost like 90% of the information are overlap, but each website probably have their own very unique, it's really useful, very specific information to the website. But that's only maybe 5% or like 10%. But why there's so much redundancy between all these websites, all these gene portals? From our from our point of view, I think that this is because of when developer develop a website, and when users start to use it and they love this website, and then user will have more requests for the different features over the time, right? But usually the development team or the resource we can put into the development is very limited. It doesn't scale over the time. So at some point, the user's request will overwhelm the development team. So the existing website cannot add as a feature user like, and then maybe other user will feel like not satisfied. Then another website probably going to be created from the community. So what we really think of this should be changing is how we can engage the community development into the gene specific portal, so that the website itself, the resource provided from the gene portal, can grow as the time along the time, and also as the user requesting the more resources. Okay, so that's why BioGPS want to emphasize the community extensibility. And also, another thing we notice is that for the existing gene portal, the developer normally defines the gene report view. For example, you go to the gene entrance gene, you can basically you will see the developer, you will see the gene report format, like a fixed format. You will find the information from this format. When you go to the new website like this, you need to learn where to find the stuff, and you probably a user don't have any control. Say, I want to maybe add another information in this portal. You cannot do that. You basically have to go to another gene portal if you find other information not included in the entrance gene portal. So we also think this is another thing we should we can make a difference. So we also want to emphasize the user customizability in BioGPS gene portal. In terms of that, I think I want to mention that the website displayed the content right now should be individually decided by the individual users, because the user probably has a different use case based on the type of studies they are using and the type of the project that they are currently working on. For example, structural biologists, excuse me. For example, structural biologists probably they are interested in knowing the gene specific, the PDB, the 3D structure related to their gene. Maybe want to know the PFAM structure for their favorite gene. Maybe geneticists are probably going to need another set of the gene portal for their favorite gene, and then also say different system biologists and the cardiovascular researchers. They probably have their own set of the resources useful for their project and then for their favorite gene. So they all have the different needs for their gene portal. So BioGPS want to build a flexible interface, which we often call a customized layout, so that different user can define their layout of a gene portal. Maybe I wanted the PDB here, maybe I wanted the PFAM here, maybe I wanted the gene wiki also in my gene report view. So that BioGPS can allow you to do this type of a customized gene portal. And I guess best way to demonstrate this is just to jump into the live demo right now, so that you can know what I'm talking about, how to customize the gene report layout in BioGPS. Okay, can you see my screen?
Yes, we can see it.
Okay. Okay, so yeah, okay, so now this is a BioGPS. Yeah, as you can see, this is like a there's like a central input box that you can paste in your favorite gene. You can paste in a list of the gene. You can for example, you get 100 gene from the genomic study. You can just paste in the not just the gene ID. You can do like gene symbol. You can do you also support the wildcard. Maybe you can like athlete list. It doesn't have to be something you need to do the transformation first. You can do the pasting your favorite identifier. I think BioGPS will always recognize it. You can also do like a gene interval. And also I want to mention at the back end of the BioGPS, all this translation of a gene ID is done by the mygene.info. Now let's say we want to do a quick search for this CDK2, CDK3. Now this returns a match gene list. Also, right now BioGPS support nine species here. And by default, it will search within the human, mouse, rat 3 species, but you can toggle the species selector here. And now suppose we want to view the CDK2. If I click CDK2, it will bring you to our default gene report, default gene layout. So in this report, you can see the different window. This window is for the expression profile for CDK2. This is all the basic information about the CDK2, all this including the function, and Go annotation, and interpro, and all the related transcripts, include all the link out to the proper resources. And down here there's a Wikipedia about a page about the CDK2. As you can see, like each resources is displayed in a window. So in BioGPS, we just call them like a plugin. Each plugin is one specific resources. You can see this window. You can move around. If you don't like this default layout, or if you want to add more stuff, you can add it to this page and reorganize it. And like for specific window, you can maximize it. You can do like a resize or this. You can essentially you can customize your different layout if you don't like. Because here there's like a quick search box. For example, if I want to have the splicing information about this gene, I can probably do a search. Then this will search in our plugin library or the resources. Right now, probably have 500, 600 of different resources you can choose from. Now, for example, I find that this one probably is useful splicing resources. I can just click. Now, this resource will be added to this current gene report. Now, if you want to do a little bit of customization of it, add it to this. Yeah, now you can view the information about splicing form about splicing graph for the CDK2 gene. Now, suppose you have all this information. I want to view the information for another gene. Now you have the gene list here from your search result. Now you can click another gene. You can see this layout is preserved, and you can see the same information but for another gene. It's very handy. If you register and also log in, you can actually save your current layout to under your profile, so that you can use it use it later. Okay, let me see if let me try to log in. Okay, let me do search. Yeah, as you can see, once I log in, it actually shows my last used layout, because I have this layout I just tested yesterday. So you can see this one probably just another one. I can customize it. Maybe I want to add the say gene wiki, add it to here. Now say if I want to here, yeah, I want to say, yeah, let's say like save as a new test BD2. Okay, save this. Now my layout is saved. Now I'm currently using my using this test BD2K layout. So as you can see, I think right now I demoed how you can customize the layout in BioGPS and how you can choose the resources from our plugin collections. Oh, I also want to show you how the current plugin library looks like. This is our plugin library. You can see that you can actually search just like we have done like for the splicing. You have all the matched one here. Okay, so now next I want to describe how the plugin actually works. Our plugin is basically user can register resources like this PubMed, and the URL basically we'll call like URL template. For example, for PubMed, you can do the query by gene symbol. Then you add the URL template using double curly braces with the symbol. Then when we render this plugin for the specific gene, we will take the gene information, find the gene symbol for this gene, and then render the real URL and display it in the plugin window I just showed it from the demo. And same for the string resources, they're using the ensemble gene as the identifier, so you can just put this and a category using the entrance gene. As you can see, different resources they're using the different identifier for their different resources. Then in BioGPS, you don't really need to remember. You don't need to get all these different identifiers. You just search for your gene and click the gene. BioGPS will do the actual rendering, find the right identifier for the different resources, and render information with the real URL under the BioGPS plugin window. And for the user to contribute, actually, it's very easy. So it's just what's the name of the resources and also what's the URL template. So that's essentially how user can register the plugin in plugin library. Because our plugin interface is simple and universally working. Right now, BioGPS user have contributed actually more than 600 different resources. This is just to give you like an overview of what are the resources in our plugin libraries. For example, this screen probably a lot of resources you are familiar with. Probably like all the genes, model organism information, gene-specific information, and this is probably a lot of related to the splicing, the genetics, the variants, all this information. This is probably related to the literature and the pattern. So this one probably related to 3D structure and protein family and functional domain, all this information. This is all the pathway, gene wiki, and all the information related to your input gene. And yeah, this is really I just also just to show you this plugin library. I want to highlight one of the plugin from our plugin library. This actually is a number one highly used plugin. It's called Expression Chart. We actually developed this. So for this one, you can actually see the expression profile for your input gene. Looks like I can hear the echo now. Oh, okay. Now it's better. Okay. So yeah, we loaded a bunch of data set, most of like a reference data set, for example, our gene atlas data set, and we also have other reference data set like NCI60, primary tumor. Besides that, right now we also have more than 2,000 geo data sets available in our data chart plugin. You can search through this geo data set and visualize and very straightforward view in expression chart plugin. So this has actually become a very handy tool for our BioGPS users, as you don't really need to go to Geo website and download the full data set and do your visualization by yourself. If you just want to check the expression profile for the specific gene for a really quick check. Specifically, we also have this so-called data set library, so that you can search through the data set, browse the different category of data set, and view their profile from the expression chart plugin from the BioGPS. And recently, we actually expanding the dataset library. We additionally loaded 7,000 array express dataset for the human mouse rat, and we also continually improve the expression chart plugin, adding the more function to this. One function I want to note here is that we added the ability to customize the chart based on factors. For example, if the data set has a different group, like a cancer versus normal, we can group the expression chart and to display the two group of a sample instead of show the expression for each sample by their sample name. We are expecting to roll out this new BioGPS data library and expression chart plugin in next month. Okay. Okay. So I have so far I have described how the BioGPS works for is a very is a major feature I described. I think we want to also summarize the success of BioGPS actually depends on what we call like positive feedback loops. We want to have first. We want to develop a really like resource sharing utility in BioGPS, so that people can register plugin and share with other people. So other people can use it. So if people all these resources people find it useful, so that we can track a number of users who likes the BioGPS, and maybe one out of this 100 users will know how to contribute back to the BioGPS if they find the known other resources currently not included. Then maybe they will contribute back to the BioGPS through our plug registering and sharing function, and hopefully by doing this we will attract more users, which in turn encourages more users to contribute to the BioGPS, so that we can hopefully we can reach this positive feedback loop. I want to next few slides I want to basically summarize how in BioGPS how we actually want to implement this positive feedback loop. As I mentioned, a utility here defined as a user customizability and simple visualization of the gene expression data. That's how people first come to BioGPS and find it's useful for their study. And over the time, BioGPS has already accumulated a critical mass of the user. This latest stats just to show you right now BioGPS have more than 8,000 registered users. I also want to mention BioGPS actually you don't really need to register to just browse the layout, preset layout, unless you want to customize your layout, save your layout, or register a plugin. You don't really have to register. Actually, a lot of users probably just use it without a registration. But of course, we do encourage people to register so that you can utilize the full power of the BioGPS. And currently we get about 1 to 2 million of page views per year, and I think this is doing really great for the biological for the web resources in the biological domain. And we're also happy to see the nice distribution between the returning visitor and the new visitor. I think this distribution represents a very healthy usage very healthy usage of the BioGPS from our user. And all this this is like just a list of top 10 organizations among the BioGPS users. So we now have the critical mass of the user, and among them, some of them actually do contribute back to the BioGPS. And one way of contributing to the BioGPS is register plugin. That's like a very explicit contribution. We have more than 160 users who have contributed over 600 plugins so far, and almost like 400 of them are publicly shared. And also, so when you register a plugin, actually you can choose whether you want to make it a public, share with others. Also, you can select it as a private if you have something, your internal resources, but you also want to register in the BioGPS, but you don't want to share with other people, that you can do that. And among all these 600 plugins is spanning more than 300 different domains. And besides this type of contribution, I also want to mention BioGPS user, like all users, when they're using the BioGPS plugin and the layout, they actually also make another type of contribution by their usage. So we also summarize the so-called layout popularity through the layout and the plugin usage, so that we can, when people search splicing, for example, search splicing from the plugin library, we display the match plugins and sort it by the layout popularity. That indicates how popular, how many people are actually using this specific plugin in their layout. So if I come to BioGPS, I want to find the splicing information, but I don't really know a good resources. I might just start with the most popular one, right? Or maybe if I know I know one of the resources, but I really don't know another one, and if we jump up to the list, I might just try this to see if there's anything new related to my favorite G. So all this plugin registration or the layout popularity, our core development team really don't need to get involved. So it will the collection of BioGPS plugin library will increase without our intervention. Okay. Yeah. So as I showed, right now we in BioGPS we can see the community participate. So all these resources can increase without our development team to putting the effort. Okay, just a quick summary so far. BioGPS as a gene-specific portal, it gives easy access to the resources you already know about, so that you can organize it in different layout. And also for the user, you can also discover the resources you don't know about, so that you can explore the new resources easily. And very likely this resource is shared by the other BioGPS user. Now next, I want to point out another different kind of use case for the BioGPS. When we develop a BioGPS and we present it to different to other collaborators. They really like it, and then we come up with another functionality called like you can call like a domain-specific portal. That means if you are you have your specific resource and want to share with other user, and we allow you to create a customized layout. We even give you like a hard URL link, for example, BioGPS Scadia, that you can use a BioGPS as an interface. People do the search, but when people click the gene, go to the gene report page, it will go to Scadia specific default layout. You can besides the customize the default layout, you can customize the gene query filter, and you can customize default data set in the expression chart view, and you can even customize the landing page. I want to give you a quick demo of this. For example, this BioGPS Scadia, that's a page we collaborated with another group who's doing the Scadia recent study. As you can see, like even we added like this logo to this specific page, and we can add some specific link relevant to Scadia search. This will goes to another link for doing the search gene by the amplitude or period that's specific for Scadia research. And then now here you do the search. It's the same search, but now if you click this, you will see the default layout becomes something called Scadia layout. So this is all the information specifically useful for the Scadia research, and people will find this is very useful if you are doing the Scadia recent research. And another one I want to show you is this. We are working with extracellular RNA community and build extra RNA specific portal for them. And for this one, as you can see, this search title becomes the search non-protein coding gene here. Now if you click this, actually the example genes becomes microRNA, and even you like search other symbol, all the search will limit it within the non-coding RNA. It won't get back won't return all the general gene. It will restrict it. We apply the gene search filter so that restrict the search to the non-coding RNA. So if you do the search, you can see all the results return back to you, match the microRNAs. If you click the gene link, you can see a different default layout right now is called extracellular RNA here. Shows the relevant information for this near 100 gene. Okay, so I think I won't try the other. There's other like a gene wiki and a primary cell atlas, and also there's a few more we have generated. Before. Well, I guess if there's a use case we can do by GPS / Scadia, like a cardiovascular specific one. Okay, so that's all I want to introduce you the BioGPS, and I think some of you are already the user of a BioGPS. If you have guys have any question and also I think we can discuss more about how we can use the BioGPS within this BD2K study. Yeah, and we can also we are also actively continue developing the BioGPS to adding more features. There's also other features, something like a gene list I haven't mentioned, but if you are a BioGPS user, you are welcome to explore all this and other useful information from useful features from BioGPS. Yeah, I want to thank all the developer, all the teams in our group, and also the past contributors. We also actually participated in the Google Summer of Code in 2012 and 2013. We recruit students working for the BioGPS, which was a very good experience for us. I think that should be it. Oh, I also, of course, I want to thank you, the BioGPS, all the BioGPS user who contributed to BioGPS resources and continue using the BioGPS as their primary gene report, gene portal needs. Okay, that should be it.
Thank you, Chunlei. That was really interesting. Do we have any questions?
Hello, this is Henning. Hi, you showed quite an extensive example on the expression profiles, and the data you import from the expression from the Array Express archive, as far as I could see. And I would just like to mention or ask, are you also using the Array Express Atlas? Because the archive is basically everything or a lot, while the Array Express Atlas is quality selected and really much better integrated sub-selection. It's a much smaller number but much higher quality data sets. So you might consider having that, if you haven't integrated it yet, to integrate this, but probably as a separate section rather than with everything else, because it's really much. Usually you don't have the problem of having not enough information. You want the filtered high quality information, and this is what I provide. So this is under just the same URL, EBI, and then /GXA. or Gene Expression Atlas.
Expression Atlas. I believe I looked at this before. Yeah, so as far as I understand it, this is like a subset of Array Express, right? Manually picked from the. Right?
It's a quality-controlled subset, but it's not just directly a subset. It's really they do much more effort there also to.
I see.
To reprocess the data, so it's not just the same as before, but a smaller set, but it's really much more.
Reprocessed. I see.
So have a look there. It should all be documented. I'm not too familiar with it, but it should all be documented, and this might be a better default entry point unless people are looking for specific experiments. But if they just want good expression data for, let's say, the standard model organisms, then this is probably the better entry point.
Yeah, I agree. And in BioGPS dataset, we have a concept called a default data set, and there's a rest of the data set. Most likely is a default data set with including gene atlas, including primary cell atlas. Those are we manually picked, and I think the data set from Expression Atlas will fit into this category. They will be highlighted. People looking for the really want to look for the expression reference. They should just pick those high quality data, and if they want the other specific one, they can just search through all the rest of the data set we load. Yeah.
Yes, exactly.
Yeah. Great. Thanks for pointing that out.
If you click on the baseline experiments or all experiments, I think this gives a quite nice overview to the right of the screen.
Right. Yeah. Oh, baseline experiment. Yeah.
Yeah, that's what I meant.
Great.
So,
I see. Yeah. Good. Yeah, and also it looks like there's a download. I see. Great. So this is like a download of all atlas. I see. So this is like for human, they combined, they made it like one, for each organism they made one reference expression profile, right?
Well, to be honest, it looks like this to me, but I am no wiser on this than you are. So as I said, I don't know the details, but that's what it looks like.
Yeah, we'll have a look. Yeah, it looks like encode and the Illumina body map. Yes. Yeah. Great. Thanks. Oh, is it is it currently connected? Yeah, I'm looking at the question from the chat box from Vincent. Is it currently connected to data set possible defined in the data discovery interface? Data discovery interface like a PRIDE, gene expression atlas, metabolite, or European genome phenotype. No, I don't think we load those data set. Currently, is that we have gene expression atlas. Gene expression atlas. Is this the EBI? This is what the atlas like Henning just mentioned, right?
Yeah. So
yeah. Okay. Yes.
May I give an answer to this?
Yeah.
I think Vincent's question is about 12 months into the future. So the data discovery interface is only planned. So unless you mean for the individual elements, but I think there is no consistent data discovery interface which links these. This is only what we plan to do. So it's a bit difficult to do that now. But you could link to each of these, and you probably already have a PRIDE link in there. You have the Array Express archive. Why don't we just discuss the Gene Expression Atlas?
Right.
Yeah, I was just making sure like if there were like plugins that already existed for them.
This is Andrew here. PRIDE and Genome Expression Atlas, those plugins both exist, but I just checked and the linking syntax is broken, so we'll fix that momentarily. Metabolites, right, is really, the point about GPS is that it's gene or protein centric.
Right.
So unless Metabolites has a query interface that is gene or protein centric, that one would be a little bit out of scope. European Genome Phenome Archive is one that I am not familiar with, so we'll look at that one. Yeah.
Okay, thank you. I guess I have another question. So to register a plugin, do you provide a link? So you showed that. Can you show that slide again?
Yeah.
For, a plugin.
This one, right?
Yeah. So yeah, you simply provide like a link, and then you put that template like symbol or whatever.
Yeah.
And is that it?
Yeah. Let me actually just show you the. Yeah, I haven't demoed that. Let me show you the plugin registration page. This is our plugin library. Right? There's a button called like a create plugin.
Yeah.
Of course, you need to log it in.
Yeah.
Now, this is like a go to the register new plugin page. This is just to give you the test plugin. Maybe this URL template. Basically, you example. Maybe like called expression / Now here, if you when you find normally you will go to this website and go to the specific page you want to show in the BioGPS plugin. Now you copy paste your URL here. Say if you have this one, maybe go to CDK2, right? You got this page. Now you basically replace the CDK2. This page using the symbol. Now here you can there's a list of the ID you can choose from here. Add a keyword. Now you can say like this symbol. Now you can, this is like a double curly braces, this placeholder for the for the actual symbol in this URL template.
Okay.
Yeah, and you can actually click the test here. Now, of course, this won't work. Yeah, you can test this.
Okay.
Yeah, no, yeah, actually, it does work. There's an example.com. So this actually, you can show that this is a real URL. It's like a CDK2, right? Now this goes to this actual page.
Okay. Yeah, that's pretty straightforward.
Yeah, it's straightforward. It's summary, and then you can, there's a permission. By default, it's public. You can choose whether it's a species specific or not, tagging. Yeah. So it's just straightforward.
Okay. Thank you.
Okay. Any other questions?
All right. I guess if there aren't any other questions, if there aren't any questions, then we'll end that presentation there. If you do have questions, feel free to email Chunlei Wu. His email is provided on the Trello board.
Yeah.
Yeah, but yeah, thank you, Chunlei, for that great presentation.
All right. Thank you.
Thank you, Chunlei.
Thank you.
Before we conclude the conference, I'll go over some quick updates. So last Tuesday we had our first BD2K PI meeting. It was great meeting everyone in person and learning about all the tools and research. We're currently processing the videos from the PI meeting, and we'll post it once it's finished. And I'll also post the slides from the presentations on the Trello BD2K general board, so you guys anyone can take a look at them. As for the DSR component, so in the past week we spoke with several collaborators, including Henning, and we talked about how we should approach the development of our APIs. Henning talked about how we should be, or how we'll try to focus on developing the data discovery interface, and as well as the expression interface for the first year of the project, and then we'll go from there. And then, do you have any training? Or
Yeah, I'm on the training side. We're currently talking to Spelman College in Atlanta as part of a outreach to increase the diversity in big data. We'll have more information on that later. Also, yeah, we're interested in the hackathon idea that was brought up over the meeting. We'll probably be participating in that.
Anyone have any comments or anything in general? Any other updates?
Oh, I have a question. So besides like a data set, like a available data set presented from the BD2K meeting, is there any other available resource, other data set available from the not presented? Maybe we should, if there's some, maybe we should also add it to the Trello page. You just add it.
Oh wait, what do you mean? Or like what other data sets?
Oh, I just wanted to check, so from the BD2K meeting, we presented the 3 cohorts, right? Data set cohort. I just want to check if there's any other available data set in this community, not included in the presentation.
Oh, okay.
This cohort, I think, actually would be really interesting too. That's one thing I wish I'd gotten a chance to talk to Carol more about. But I was just going to email her, but that could be a broader interest.
Right.
Okay. We'll look more into that. And yeah, if I find anything, I'll try to post it on the Trello board.
Great.
Any other questions? Okay, we'll conclude this meeting early. So me and Brian will stay online for any questions or comments till 10 a.m. But yeah, thank you for coming to the conference, and thank you, Chunlei Wu, for that great presentation.
You're welcome. Thank you.
Thank you.
Thank you.
So one quick question. Pepe, are you on the line? Is Pepe on the line? Not any.
I don't think so.
Oh, okay. Okay, thank you.
Thank you.
Thanks.
Great.